For today, we're going to be talking about microbial genetics. If you've taken any kind of biology course up until now, you'll probably know some of these important definitions, which include the genome, which is all of the DNA that's present in a cell or a virus, a genotype, which is the specific set of genes that an organism has, and the phenotype, which is how genes tend to manifest. It's the observable characteristics of an organism. So things like physical characteristics like eye or hair color are designated by your genetic makeup. It's also important to acknowledge the central dogma, which states that DNA is always transcribed into RNA, and then RNA gets translated into protein. It always proceeds in a singular direction. You will see here, however, an example of a special scenario where RNA can be made back into DNA. And we have talked about a few examples of this in viruses that have special enzymes that permit them to do this. But most organisms, whether they're eukaryotic or prokaryotic, always run from DNA to RNA to protein. It wasn't always known that DNA was the material within the cell that contained genetic information. There were a lot of early debates around whether or not it was actually contained within proteins, because proteins are made up of more complex structures. If you consider that it takes 20 different amino acids to comprise most of the proteins within the human body, and only 4 nucleotides in order to make up all of the different genes and DNA that we can possibly have, you would automatically tend to lean toward protein. A number of experiments were performed, and eventually it was proven that DNA did house all of the genetic information. So one important thing to try to get straight off the bat here is that genetic information tends to flow in 2 directions in the cell, and we discussed DNA in the context of 2 major processes. The first is DNA replication. And this is when we have DNA that has to be doubled to transfer to a new generation of cells. That is different from gene expression or DNA transcription, where genes get transcribed into mRNA and from there get translated into proteins and expressed as such. So if you've ever had a difficult time determining which process we're talking about or feeling like a lot of these processes overlap, first identifying whether we're talking about replication or transcription and translation will hopefully help to get some of these things straight in your minds. I've included a couple of diagrams here just to kind of help you get a visual for these types of things. And again, your replication shown here is just the doubling of the DNA, typically during the cell cycle prior to cell division, right? Because each of the daughter cells is going to need its own new copy of DNA. You can see that over here in this diagram as well. It's making a new copy of the DNA from the parental DNA. Also, the transcription and translation steps, where we have transcription, which is essentially the formation of mRNA from the DNA template. And if you get confused between transcription and translation, think of transcription as DNA and RNA are the same language, right? They're both nucleotides. Whereas translation of RNA into protein forces you to convert that to a different type of molecule. So you're going from nucleic acids to amino acids. Transcribing is the same language, translating is a different one. And you can see here that process is shown. Over in this diagram, the same idea here. We've got our different types of RNA that are produced from transcription, from genes, and then from there, these different types of molecules can be read and formed into proteins throughout the process of translation. The DNA structure is going to be the same no matter whether or not you are eukaryotic or prokaryotic. You're always going to have the 4 different nucleotides that are going to fall within the 2 different categories. You have your pyrimidines, which are cytosine and thymine, and your purines, which are adenine and guanine. Adenine, of course, always binds with thymine, and there are 2 hydrogen bonds that form between those, and guanine is always going to bind to cytosine, and that's going to be with 3 hydrogen bonds. Now, once again, I'm not ever going to ask you to be drawing any of these molecules out at the molecular structure, but what I would like you to know is what pairs with what, and maybe some of the details on this slide, like how many hydrogens are bonded between different base pairs, and that sort of thing. This is taking the DNA structure a little bit further. So along with our nitrogenous bases, of course, we have pentose sugars and phosphates that make up the rest of the DNA molecules and the DNA ladder. The pentose sugar is going to vary depending on whether we're talking about DNA or RNA. in DNA, you're going to find that it is deoxyribose, because if you take a look at the structure here, you'll see that there is an oxygen missing or deoxygenated. It's missing an oxygen atom, whereas over here you can see in ribose, which is found in RNA, you do have that additional oxygen atom present. The other big difference between DNA and RNA is the presence of uracil. There should be a U here. I'm not sure where that's gone, but the uracil is only found in RNA and actually replaces thymine in that case. So in that particular type of molecule, adenine is going to bind to uracil versus adenine binding to thymine in DNA. So when we put it all together, these are some great diagrams to show what it actually looks like. So you've probably heard DNA referred to as a ladder-like structure because each of the base pairs that are hydrogen bonded form the rungs that ascend all along the molecule. You can see that here depicted in a little bit more of a schematic form, and on the right here you can see some three-dimensional representation of these to show it in a little bit more detail. But hopefully at this point this is a little bit of a review for you. We talk about genes pretty often, but what is a gene? It's a segment of DNA that's going to code for a specific protein via mRNA, or it can be tRNA or rRNA, respectively. In eukaryotes, genes tend to be split along the genome with two or more coding regions, with what are called non-coding regions in between. The coding sequences that actually translate directly into mRNA and form proteins are called exons, and the intermittent non-coding sequences are called introns. When we actually undergo DNA transcription, the intron sequences are transcribed along with the exons into what's called the primary transcript. So the whole section, whether or not it contains coding or non-coding information, is going to get transcribed into mRNA. And then after that, the mRNA gets modified through excision or cutting for non-coding regions to remove them. Generally speaking, prokaryotic cells do not contain any intron regions. Their DNA is comprised primarily of exons. There are some exceptions, but the majority do not have intron regions included. We can look at this in more detail here. You can see here on the left we have an example of prokaryotic cells where we have in our DNA gene A and gene B. In transcription, you'll see that there are no non-coding regions, so coding region A, coding region B, and when we go through the translation process with our ribosomes, we end up with two different products from that translation step. When we take a look at eukaryotic cells, we'll see that if this is an entire gene that has several coding regions here in the form of exons, in between those are intron regions. When we do the transcription step, we end up with a transcript that will contain both the exon information and the introns as well. Following that, we have the RNA processing that I mentioned, where you have those intron regions removed through excision. At the end of that, you end up with what's called mature mRNA, which can then be directly translated into protein. And you can see down here that even if a single gene has several sections that are divided up by introns, at the end of the day, we still end up with a functional protein product at the end that can then go off to start doing its work. Now, we've learned a lot about eukaryotic DNA over the years, but let's have a look here at what bacterial DNA looks like. It's going to be circular with what are called coil and supercoil structures. This is achieved through nicking DNA, so causing little cuts and clips in it to create torsion, and DNA gyrase, which you've probably heard of before, is expressed in bacteria and helps to create these supercoils in DNA. So what you can see in this diagram here is that if you do have nicking made in the DNA, that's going to allow for that material to be twisted in on itself and wound up to become more compact and compressed, versus the large splay of DNA just hanging out without kind of being self-contained. Because don't forget, we're talking about cells that do not have a nuclear compartment. So all of the DNA is going to be in the cytoplasm as it is, but you still don't want to just have it taking up all the room in the cytoplasm. So you do still want to have that supercoiling effect to make sure that DNA is kind of tucked away in one area in that nucleoid region. If a cell cannot supercoil their DNA, they're going to have a very difficult time packing it into the cytoplasm. And so you'll see that a lot of different types of interventions that we have in terms of chemotherapeutics actually target DNA gyrase so that that supercoiling event won't be allowed to proceed. A little jump over to archaea here. Archaea will actually have more similarity to eukaryotic DNA than bacterial DNA. This is mainly due to the presence of histones. Histones are big proteins that help DNA to coil around and become organized. So archaeal DNA does not get supercoiled, but instead mimics the histone system that we would normally see in eukaryotic cells. And you can see here that as the DNA is coiled around these histone proteins, it's going to form a larger structure that's referred to as a nucleosome. Having them arranged in this type of orientation also helps to protect them from nucleases, which, as you can imagine, are enzymes that cut up DNA. Now, a long time ago, we did talk about how mitochondria and chloroplasts are extremely similar to prokaryotes in terms of their own unique structure and function compared to other organelles. Chloroplasts and mitochondria both contain their own genes, and we're going to take a look at those here just for a moment. Chloroplast genomes are also very similar to what we see in bacteria. They are circular, and they also contain a set of unique genes that are not coded for in the cellular DNA. They're very specialized and tend only to code for proteins that are involved in photosynthesis and autotrophy, as well as rRNAs and tRNAs. In addition, you can also find some genes that are similar to those found in different bacterial species, like those that are necessary for cell division, but a key difference here is that chloroplast DNA does contain introns, so it does more closely resemble eukaryotic DNA with those intron regions interspersed, versus not having them like what we see in prokaryotes. In mitochondria, you'll still see a similarly structured DNA type, but you'll see less complexity. So you'll see only about 13 genes in human mitochondria that will code mostly for things like the proteins that are involved in oxidative phosphorylation, as well as rRNAs and tRNAs. So this is just a basic schematic, and of course you don't need to memorize any of this, but just showing you here that some of the different complexes that are involved in the electron transport chain here are coded for directly in the mitochondrial genome. We're going to shift gears now and start our discussion about DNA replication. And again, this is the process by which a cell is going to copy its DNA. It's going to be unique depending on whether or not we're talking about eukaryotic cells versus prokaryotic. And this is primarily due to the structure of the DNA and the differences that we see between these two types of organisms. Taking a step back here, though, originally we had 3 different proposed models for DNA replication. There was what was called conservative replication, semi-conservative, or dispersive. So when we look at conservative DNA replication, this would imply that DNA gets replicated and parental strands reattach to one another following the event. So let's take a look at this here. We've got our parent cell DNA. We have the first replication where the new replicated DNA is its own thing and the parental DNA reanneals after the process is done. And as a result of this, we're always going to end up with brand new strands, 2 brand new strands with every new replication. Semi-conservative addresses that both strands separate and stay separated after DNA replication. So in this case, we have the 2 parent strands that each act as their own templates to form new DNA strands, and then those go on to form new DNA along the way. So semi-conservative meaning that half is conserved and half goes on to be new DNA in that initial replication step. The third here is dispersive, where each strand would contain bits and pieces of new and old DNA willy-nilly. So if you take a peek here, you can see that the parental DNA is here shown in dark blue, and then this would imply that different portions of this DNA get split during the replication process to eventually form almost like a large hybridized structure versus something that's a little bit more streamlined. If you haven't been asleep at the wheel during the last few courses, you will know that the winner is actually semi-conservative DNA replication, where we do see the DNA from the parent acting as a template for new DNA on either side. In prokaryotic DNA replication, this is still true, except for the fact that we're going to have all of this replication beginning at what's called the origin. The origin of replication is shown here. We have 2 replication forks that are going to form at the origin, and they're going to move outward. So you're going to have one that's going to move kind of counterclockwise here, and one that's going to move in the clockwise direction. So you can see here as the proteins bind and start to make new DNA from the template strands in one direction, you're going to have DNA being formed here, and you're going to also see the same process happening in the opposite direction. By the end, you'll have 2 almost nearly complete genomes, which can then be eventually split off at the terminal site. And then you end up with two brand new semi-conservatively replicated genomes. This diagram here shows a representation of E. coli's chromosome during replication, and it actually does show those replication forks moving away from that replication origin. Because eukaryotic DNA is not circular and it's linear, what we can actually see in these cases are multiple origins of replication instead, where you can have many different sites along a very long piece of DNA that can actually initiate replication. Because eukaryotic DNA tends to be a lot more expansive and larger, you do need this for the speed. Having multiple origins of replication allows for faster replication of these much longer and larger molecules. This is shown well here in this diagram, of course, with replication forks going in either direction, but you can also see this here shown in some electron microscopy as well. When we look at the actual machinery or enzyme complement involved in DNA replication, archaea and eukaryotes are going to have very similar machinery, whereas bacteria are going to have something that is a little bit different. The overall process in all three are the same, but the components that do the work are different. DNA synthesis or replication is catalyzed by the enzyme DNA polymerase, and all DNA polymerases synthesize DNA in the 5' to 3' direction. When we talk about genetic exchange of genes, prokaryotes typically divide in an asexual fashion, where they go through this DNA replication step and then eventually divide into two new daughter cells. But there is still a need for the diversification at the level of the gene to produce genetic variability within a population. So what we have are several kinds of gene transfer. There can be horizontal gene transfer, like transformation, where bacteria will take up DNA from the local environment. These are typically linear segments of chromosomal DNA or plasmids, which we're going to talk about as well. There's also transduction, where DNA gets transferred to different bacteria through viruses. Typically, this is done through bacteriophages, which we talked about a long while back. And these are viruses that actually infect bacteria by inserting their DNA. And sometimes DNA from other bacteria can get inserted into a new one through this process. Then there's conjugation. Conjugation occurs by direct cell-to-cell contact and is typically intentional. This greatly enhances genetic variability, but also helps with adaptation. Conjugation usually involves plasmid exchange, and this is done through a sex pilus. Plasmid DNA tends to be extraneous DNA, so it doesn't code for anything that is essential and is not typically housed inside of the bacterial genome. It's a separate set of genes that's on its own little circular piece of DNA that is outside of the normal chromosomal DNA. Just because the DNA is not essential, it doesn't mean that it's not beneficial, because some of these genes can be great for adapting bacteria to their environments. Some great examples of these contain antibiotic resistance proteins that can get passed from one cell to the other to give them the ability to resist antibiotics or any other environmental stressor that may be present. In addition, plasmids are replicated independently of chromosomal DNA, though the process is quite similar. It's done through what's called the rolling circle method. In this case, we have one strand of the DNA that gets nicked, which will expose the hydroxyl end, which will act as a primer to attach new nucleotides. So you can see here in this diagram, we have a nick that's going to occur. We're going to have an opening here and a little replication fork that's going to form. And as this gets elongated, you'll see at the end, we end up with a brand new copy of the plasmid over time, which can then be sealed up. You might be wondering why this linear phase is even required, and this is because this DNA is being replicated and passed to a new cell, and it has to go through that sex pilus. The pilus is just a small little tube that couldn't pass something as large and bulky as an intact plasmid, so as that replication is occurring, this little linear piece is squeezing through into the other cell. Now we're going to switch over to a conversation about transcription and translation and get into some talk about gene structure. Because apart from just being a simple method to pass on the genetic information of one generation to the next, a lot of genes are used to actually make functional products. All of those enzymes that we've talked about with respect to metabolism or other cellular function are coded for in DNA. We've mentioned already that rRNA and tRNA are both things that can be coded for in DNA, but mRNA or polypeptides, and this is where most of our focus is going to be for today. And again, keep in mind that central dogma where DNA is going to get transcribed to the same language, right? We're sticking with nucleotides into mRNA. And after that, mRNA is going to be translated into protein or amino acids. Now, bringing these concepts back around again, genes do not overlap. They are all going to be independently arranged on genetic material. And again, in eukaryotes, we do have the presence of introns, which are non-coding segments that are squeezed within a gene or even between different ones. Prokaryotic cells tend to not have interruptions in between. And if we look at this genome for E. coli, you'll see that every little bit of this circle is occupied by something. There are no portions of this that are not coding for anything. So there are no intron regions in this particular genome. So DNA has to be transcribed to mRNA before it can be translated to protein. All DNA is double-stranded, no matter what type of cell you are, whether you're eukaryotic or prokaryotic. So you'll have your coding strand and your template strand. And after transcription, you'll see that you get your mRNA from that template strand. If you are astute, you'll notice that the transcript here is exactly the same as the coding strand, except it's mRNA because it contains uracil and not thymine instead. So that's something to think about if you're ever asked on a test to do a question about what your coding strand or your template strand or what your transcript would look like. Your coding strand and your transcript are always going to be the same. Again, differential based on the type of nucleotide that's been incorporated. The gene itself is broken down into 5 different regions. And when we talk about upstream and downstream, think of upstream as the beginning or where transcription is going to start, and downstream being toward the end of that or where transcription is going to end. At the very top of the gene is going to be what's called your promoter. It's the site where RNA polymerase is going to be able to detect and attach, notifying that is the beginning of the gene. That portion, that promoter region, acts more as a docking site. So it's not part of the gene product at the end, but rather just a landing bay for that type of machinery to come in and attach. We also have what's called the leader. This is the Shine-Dalgarno sequence, where rRNA is going to bind and transcription is going to begin. This is another component that does not get translated, or in other words, is not incorporated into that final product. We have the coding region, which will always start with our start codon, TAC, whereas mRNA is going to have AUG, which always codes for the amino acid N-formyl methionine. After that, we have the trailer, which is needed for the proper expression of the coding region, which is also not translated. And then last but not least, the terminator sequence, which will cease RNA polymerase's activity and cause it to fall off of the template strand. So a way to think about this in simpler terms is you have a way for the machinery to attach. You have a buffer region prior to beginning the actual transcription of the coding region, just in case something goes wrong, right? You have a little bit of a pillow on one end. You have your big coding region, which is actually the part that gets transcribed and translated. And then at the end, you have your trailer, which will allow for a slow shutdown of the process, and then the terminator, which stops it. If you look at what actually ends up in our transcript here in our mRNA, you've got your leader, you've got your coding region, and you've got your trailer. Your terminator and your promoter are not included. The transcription of genes is something that's not necessarily needed all the time, right? You're not going to have your cells just constantly making gene transcripts and translating them to make a bunch of proteins. There is some degree of turning these processes on and off when they're required. And as you might imagine, this is very highly regulated. You can have transcription regulated positively or negatively. Are sections of that promoter region where proteins called repressors can bind that will physically block the binding of RNA polymerase and will not allow transcription to occur. Or you can have activator binding sites, which will enhance transcription or cause it to occur more frequently. There are 3 processes in bacterial transcription, which include initiation, elongation, and termination. Initiation is where you're going to have the polymerase and its complex proteins binding to the promoter region. There's elongation, where you're going to have mRNA synthesis by the RNA polymerase and its complex. And finally, termination, where you're going to have the interaction of RNA polymerase with the trailer and its components to halt that process. Because transcription only requires one strand of DNA to be used, this is what will help to make it much less complicated. You'll see here in this case, we have our RNA polymerase that's going to be moving along and adding new nucleotides in a single direction. We don't have to worry about a lagging strand. We don't have to worry about primers, nothing like that. We're only working off of one strand to produce that mRNA transcript. Translation here should hopefully be a bit of a review, but it is a process of reading mRNA in order to create proteins. This is done in a reading frame in groups of 3 nucleotides that are called codons, and it also has a form of punctuation, so starts and stops. The AUG, which we mentioned, is our start codon, which initiates translation, and then there are several different kinds of stop codons, UGA, UAG, and UAA, and these are going to signal that translation is due to stop. Now, these are just a couple of examples, but if we look at the DNA, and then we look at the mRNA that's produced following transcription, the translation step is going to read this mRNA 3 base pairs at a time, or in a codon form, and each unique set of 3 corresponds to a specific amino acid. So AUG is methionine, CCA is proline, UAC is tyrosine, UGG is tryptophan, and so on. You can see the same example over here where different codons are going to get produced depending on the unique structure of different proteins and different gene transcripts. The codons of mRNA are going to be read by ribosomes, and tRNA molecules, or the transfer RNA molecules, are going to change information from nucleic acids into amino acids. So the ribosomes are bound to the mRNA, and as they're moving along, they have a couple of different sites that are going to be reading codons in that 3 nucleotide formation. The tRNAs are going to carry specific amino acids to the ribosomes, which is going to add a new amino acid to a growing chain. So you'll see in this example, we have at this site a tRNA that was once bound to an amino acid that's leaving. So that's on the way out. Right here at UUU, we have a tRNA molecule that corresponds and has the corresponding amino acid conjugated to it that's going to come in. So that's going to add on to the length of the chain that's growing. It's growing from this end, so the end that is bound here. The next one in line, AGC, is going to correspond to a tRNA that is UCG, and its amino acid here is going to have this chain added onto it. It's going to shift over and continue the process. This one will leave, a new one will come in, and so on and so forth. And as that happens, that protein is going to be elongated along the way. At the initiation of translation, the 2 ribosomal subunits, which are different sizes, 50S and 30S, are just hanging out in the cytoplasm. And when the translation process is initiated, those 2 will form and join up around that mRNA molecule. In order to increase this process, many different ribosomes can bind a single mRNA strand and start working on it. And this polyribosome formation is going to make it so that we have increased protein synthesis in a shorter amount of time. Prokaryotes can also start translation prior to transcription being terminated, which is something that doesn't really happen in eukaryotes because of that extra processing that's required with intron removal. So because all of the genes that are coming out of prokaryotic cells are ready to be translated, there is no need to wait. This is just showing the general structure of our transfer RNA, where we have one end that's bound to an amino acid that corresponds to the codon that it will bind to in the mRNA sequence. When translation is initiated, we have the small ribosomal subunit that's going to come in and bind to that start codon. In bacteria, that's going to be N-formal methional tRNA. In archaea, that's going to just be regular old methional tRNA. The structure is going to move down the mRNA until it reaches that start codon, and then the large subunit will come in and bind, creating an active ribosome, and then that process that we just discussed can begin. Ribosomes have 3 sites that are important for elongation. We have the A site, or the amino acyl site, the P site, or the peptidyl site, and then the E site, which is the exit site. At the A site, this is where complementary tRNA molecules are going to be coming into the ribosome and binding. We have the P site, which is where the transpeptidation reaction is going to be occurring. Transpeptidation means linking of 2 peptides together, like we talked about, the passing of that chain over to the next tRNA and joining. That's done by peptidyl transferase. And then after that amino acid is removed, as the ribosome is moving along, There's the E site, which will facilitate the exit of the tRNA that's now lacking an amino acid because it's donated it to the chain. So you can see this in more detail here, where we've got the P site that has that tRNA molecule with its amino acid already added on. We have a new tRNA coming in to bind at the A site, again corresponding to the codon in the mRNA sequence that'll come in and bind. We have the transfer of this chain over to the tRNA and its respective amino acid in the A site. As the ribosome moves along, you'll see now this guy in the P site has lost its amino acid and it gets ejected. And then that other tRNA that was in the A site is now in the P site, and the process repeats and repeats as that protein becomes elongated over time. Finally, there's the termination step, and that's going to occur at one of 3 stop codons that we already discussed. When that gets to the A site of the ribosome, that's the trigger for that process to halt. No amino acyl tRNA has a complementary sequence to the stop codons, and as a result of that, release factors will bind instead, causing the ribosomes to fall apart and separate. So hopefully this shines a little bit of light on some processes that we see in prokaryotes that we have not yet looked at, since we primarily tend to focus on eukaryotic cells. Thank you very much for your attention today. I'll leave it there for now. And of course, please let me know if you do have any questions. You can reach me at laurar@nipissingU.ca, and have a great rest of your day. Bye for now.